this is part 3 of the Metabolic Pathways lectures, and we left off with photosynthesis. We learned that photosynthesis occurs in plants. It occurs in organelles called chloroplasts, and that it includes 2 stages, the light-dependent reactions and the Calvin cycle reactions. So we're going to begin with light-dependent reactions and discuss those reactions in more detail. Since they're light-dependent reactions, of course, these are the reactions that involve sunlight. The different pigments in the plant, such as chlorophyll A, chlorophyll B, carotene, there's xanthophylls, there's just different pigments in different plants. But definitely the 2 chlorophyll pigments, chlorophyll A and chlorophyll B, which are 2 different shades of green. These pigments absorb sunlight energy, and then a photon of light energy strikes the antenna pigments. They're called antenna pigments because they absorb light energy. Strikes the antenna pigments of photosystem 2, which is located within the membrane of the thylakoid, to initiate photosynthesis. The energy travels to the reaction center chlorophyll A molecule, then to the electron transport chain, which pumps hydrogen ions. Now, where did those hydrogen ions come from? They came from water. Into the thylakoid, interior of the thylakoid. Remember, a thylakoid's kind of like a flat disc. So in the membrane of the thylakoid, that's where the photosystem 2 is located, which contains the different pigments. And then the interior of the thylakoid is where the hydrogen ions will build up. So they will build up a high concentration in the interior of the thylakoid. So when they come into contact with ATP synthase, which is a channel protein that allows the hydrogen ions to move down their concentration gradient. Since they've built up such a high concentration inside the thylakoid, they're going to move out of the thylakoid through ATP synthase. And this process is called chemiosmosis. And that flow of hydrogen ions through ATP synthase provides the energy to phosphorylate ADP and produce ATP. So ATP is produced during the light-dependent reactions. And then in a second group, with a second group of antenna pigment molecules, also located on the thylakoid membrane, these are called photosystem 1. Photosystem 1 pigment molecules absorb a second photon of light energy, and this results in the formation of NADPH. So basically, the 2 products of the light-dependent reactions are ATP and NADPH, and these 2 chemicals power the light-independent or Calvin cycle reactions. So let's talk about what a photosystem is specifically. We have photosystem 2 and photosystem 1 during the light-dependent reactions. And what a photosystem is consists of a light harvesting complex and a reaction center. So the antenna pigment molecules and the reaction center, which contains a molecule of chlorophyll A. Pigments in the light harvesting complex pass light energy to 2 special chlorophyll A molecules in the reaction center. The light excites an electron from the chlorophyll pair, which passes to the primary electron acceptor. So it's going to, the energy is going to pass from pigments to the chlorophyll A the chlorophyll A molecule found in the reaction center. Then that energy is going to pass to a primary electron acceptor, and then it is going to be used to produce ATP and NADPH. This is photosystem 2, and this is photosystem 1. And you can see the pigment molecules located in each photosystem, which absorb sunlight. And that energy finally reaches the reaction center, which contains 2 chlorophyll A molecules. The energy in the form of an electron then travels to a primary electron acceptor, and then that energy is going to be used to produce ATP. The only thing I want to show you is that photosystem 2 splits a water molecule into oxygen and hydrogen ions and electrons. And these electrons from the water molecule replace the ones that are lost from the photosystem. And oxygen is released as a waste product, right? This is also the process where oxygen is released to the atmosphere. This is a blow-up image of photosystem 2 and photosystem 1. Photosystem 2 just comes first, and then photosystem 1 comes second. I'm sure there's a reason why, and I've never bothered to learn, but if any of you want to Google it, feel free. But this is how the process works. This is the way the process works. You have to remember that in order to produce ATP by oxidative phosphorylation, we have to have hydrogen ions and we have to have electrons. Okay, so let's go through the process. The splitting of a water molecule releases electrons and hydrogen ions. And hydrogen ions are, okay, the electrons, once they leave the electron acceptor molecule, they end up being passed through these carrier molecules, which this is the electron transport chain. And as the electron is passed from carrier to carrier, that energy is used to pump hydrogen ions into the interior of the thylakoid, where the concentration of hydrogen ions builds and builds. So the concentration of hydrogen ions is high, and what would happen by diffusion is if hydrogen had a way out of the thylakoid, it would leave the thylakoid by diffusion because molecules move from their high concentration to their low concentration. So ATP synthase is the way out for the hydrogen ions. They diffuse down their concentration gradient through this ATP synthase molecule. And this flow of charge, flow of hydrogen ions, generates the energy to add phosphate to ADP, producing ATP. And then that electron that leaves the electron transport system ends up in photosystem 1. And the energy from that electron is used to produce NADPH. So we have the production of ATP and NADPH during the light-dependent reactions. Now we come to the light-independent reactions, or the Calvin cycle. And this is the process where ATP and NADH are used to produce carbohydrate molecules such as glucose. I want to go back to one thing though. Make sure that you understand oxygen is produced during the light reactions as a result of the splitting of water. Just remember that, okay? And the final electron acceptor, where that electron ends up, is in this NADPH. The hydrogen atom in the NADPH contains the final electron. So that's where it ends up it goes through all this whole process and ends up in NADPH. And then water is split to replace the electron that was taken up by NADPH. So sorry about that. I just wanted to make it clear because it can be really confusing. So the Calvin cycle is the reactions that do not require light. They don't have to occur in darkness, but they just don't require light. So what's happening in the Calvin cycle is that carbon dioxide is used to produce a molecule called G3P, or sometimes you will see it written as GA3P. They're the same thing. It just depends on different ways that they're written. There's different people abbreviate that molecule different ways. The name of it is actually glyceraldehyde 3-phosphate, but you don't have to know that. Notice that during the Calvin cycle, carbon dioxide is used to produce G3P, which can be then used to produce sugar. G3P is a molecule that contains 3 carbon atoms. So 3 carbon dioxides are needed to produce 1 G3P. And during this process, ATP and NADPH are used as the energy source to produce the G3P and the sugar. But G3P is also used, some of the G3P is used to regenerate RUBP. And that's ribulose biphosphate. And that molecule is needed to keep the Calvin cycle going. That has to be regenerated so the Calvin cycle will keep going. So 5 molecules of G3P are given for every 1 molecule that's used to make sugar. And here you see some numbers, so you can maybe understand it a little bit better. Every time 6 molecules of GA3P or G3P are produced, every time 6 are produced, each one of them has 3 carbons, okay? As you can see here, there's 1, 2, 3 carbons in G3P. Well, 1 molecule of G3P is used to produce glucose. Of course, glucose is going to need 2 molecules of G3P, so it only produces half a molecule of glucose. 5 G3Ps or GA3Ps are used to produce the RUBP, the ribulose biphosphate, which is a 6-carbon molecule. It's a 5-carbon molecule. Okay, so when it combines with the carbon dioxide, it produces a 6-carbon molecule, which is immediately split into 2 molecules called 3PGA. Anyway, it's split into 2 molecules containing 3 carbons each, which you see here. There's actually 2 of these molecules that are produced. And that's the first step. It's called carbon fixation. It's when carbon dioxide is added to RUBP, and that 6-carbon molecule is split into 2 3-carbon molecules. That is called carbon fixation. It's when carbon dioxide is converted into an organic molecule. And then in stage 2, we have reduction of this molecule. It's reduced, and that's when the G3P is produced, GA3P. And then we have the regeneration of RUBP, which of course 5 molecules of GA3P are used for every 1 molecule that's used to produce glucose. And in the next lecture, we're going to move on to cellular respiration and talk about that a little bit more.